Good evening. I think I can start the session. People will be joining us later, I think. Thank you for joining, Shashank. So I'll start discussing the questions with whoever joins. Uh, let's see how many people join. So today's week six content is about what and all you guys have studied here. It is about transgenic plants and what else you guys have studied. Mostly transformation, right? Transformation based on agrobacterium and those stuff. Okay. So let's first go to the questions. Along with discussing the answers for the question, we will be discussing in detail about each. Foreign gene that is present in genetically modified organism is called a dash. So yes, transgene. So what do we mean by transgene? Transgene is what actually? Simple definition of transgene. Foreign gene, yeah, it is just as it's given in the question. It is any foreign gene which is incorporated by gene transformation methodologies inside any organism or cell. Those things we will call it as genetically modified organism. So, in case of transgenic plants, so in what ways we can obtain transgenic plants? There are certain um, methods or different ways from which you can obtain a transgenic plant. So, can you name something? Like, what are the techniques which will be used for making a plant a transgenic one? Um, you can think about hybridization. It is one way of doing a transgenic plant. The other is like you can use any mutagenic agents, both physical and chemical. So mutagenesis you can do. What else you can do? You can do. You can administer somatic hybridization, and also by using hairy root culture, so we can write plant tissue culture using hairy roots. Yes, transformation via vectors like yeah, you are right, Sasank. We can do that also. Give me a minute. So transformation, how do we do transformation in plant cells, and how do you generally do it in your microbial cells? How do we generally do it? So, so how do you do transformation in generally in an E. coli or something? How do people do transformation? So they generally have a prepare a competent cell where they will make holes in the cell surface, and then they will have a multiple heat and cold cycles and adjust the by the use of some chemical reagents they will adjust the pore whether to open it or close it. So when they introduce the new DNA inside, it will go inside and close. So now we can call it as a transformed uh, cell. If it is E. coli DH5 alpha something like that, this is the transformed cell. So so this is how they do it in microbial cell system. So how do we do plant transformation? So few techniques I have explained there, how a transgene is going to be incorporated into a plant system. So genetic transformation, can you name any one technique where most commonly used for transforming a plant cell? Any idea about how do you transform a plant cell? You would have studied in this lecture about different techniques which can be used for transforming a plant cell into a transgene containing one. No idea? No idea. So let's go back. Mutagenesis, you will just, yes, Sashank, you're right. So agrobacterium mediated. In both ways you can do, you can use strain agrobacterium rhizogenes or isobacterium rhizogenes. It is the same strain. That will yield to a hairy root culture, and you can also use agrobacterium tumefaciens that will yield a tumorogenic callus, which will have transformed tumor like callus form. So that you can do. You can remove the unwanted genes and then add the necessary gene of interest into the in case of tDNA you can insert there, and then both in case of RNA and TA plasmid it will be transfected inside your plant cell. The outcome will be phenotypically selected by the uh, by absorbing hairy root in case of Rhizospecterium rhizogenes or Agrobacterium rhizogenes, and tumor formation in case of tumor. So mutagenesis, what are the physical and chemical mutagens? Can you define something? Generally, even in microbial system, how do they do mutagenesis? They will try to do random mutation. So generally, people use targeted mutagenesis or non-targeted mutagenesis, random mutations or targeted mutations. Targeted. Yes, so if you want a particular gene to be overexpressed, you will try with different physical or chemical mutagenic agents, right? So physical in the sense you can name UV radiation, IR radiation, gamma radiation, something like that. And chemicals, you can there are certain mutagenic chemicals. For example, not able to recollect things. So there are some reagents which can cause mutagenesis, but Targeted mutagenesis, that's our aim, but not quite often we will observe these mutations happening. So somatic hybridization, you remember we were discussing about uh, somatic embryogenesis, right? Similar way, two different protoplasts. What is protoplast? So what are somatic cells? Yes, Shashank, it is cell wall. Yeah. Um, so these are somatic cells. They are not gametogenic, and the cells after removing your cell wall, it is called protoplast. And when both somatically originated cells are being fused together, you will get a hybridized cell. So in case if you are looking into people do right potato, and I guess there is something called tomato. So they do potato and tomato they fuse together and make it as tomato. It has both the properties, both the fruits properties, like veggies properties. So similarly, if you want to incorporate characteristic of one particular plant cell into the other, just remove the cell wall, uh, putting it in whatever based on osmotic tolerance, put it in their media, and then the cells will get fused together. You now will get a new cell with the genetic makeup altered. So, of course, plant tissue culture, as you guys explained, the hairy roots and tumorigenesis using agrobacterial strains. So, the second question is, DAS provides the site of binding of RNA polymerase, and it's involved in the initiation of transcription. What would be the answer for this? Promoter. Yes. So, promoter is the site where your RNA polymerase will bind, after which transcription starts. So, what is exon then? Exons and introns? Yes. Which one is coding and which one is non-coding, Sashank? Yes, exon is the coding region and your intron is non-coding region. This introns will be sliced or spliced when during which process? After transcription or before transcription? After transcription, it will be sliced post. It is a yes during RNA processing, right, Sashank? You're right. So, what are the other things you when you design a vector or something? So, what are the other things you will have it in the entire what do you call it? So, when you design a template or DNA or something, let's call it as a gene, your gene construct we will call it. So, what are the things you will have it in your So let's consider this as your gene construct. And what are the things you say? For example, your promoter will be there. What else will be there? Many exons will be there. Exons, introns. Say for example, exon is there, intron is there. Okay, we will write stop codon also. Stop codon. What else will be there? Promoter, exon, intron, UTRs, leader and enhancer. Okay. What is the order of the sequence? There is an order, right? You cannot just randomly keep your like, like leader sequence somewhere, enhancer somewhere else. We all know that promoter is the initiation site of transcription. So where it will be placed generally at the initial stage, right? So after promoter, what will be kept actually? After promoter, what will happen? Your promoter will bind your RNA polymerase. After which, what you will keep? Okay, after which, according to, okay, first is promoter, then your enhancer or silencer also we can. After which, what we can keep? Okay, so RNA polymerase is going to just bind in promoter, and where is the transcription initiation happens? Like, okay, initiation happens here. Where actually is your transcription initiation site is there? So there should be a transcriptional start site, right? What is that? Start codon. Yes, right. It will come after your enhancer and silencer, whatever you keep. After that, your start codon will come. After started your start codon, what will happen then? You can keep your leader sequence. Then comes your exons, then introns, and then the stop codon comes. And there will be some untranslated regions which Sachang calls like UTRs. UTRs. After your, say for example, your stop codon is here means at the end what will be there? What will be there here at the end? Start codon is there, stop codon is there. After stop codon, what will be there? It is necessary for your, give me a minute. You think and answer, take a minute. So what will be the answer at the end after your stop codon? Something which is necessary for your, which is related to a post-translational modification. Yes. Shashank is right. It is poly A sequences or poly A tail. Um, okay, thank you for answering all this, both Asta and Shashank. So we have some new people joining over now. Okay, so let's go to the next question. So we were just discussing about the things in the previous slide. So what is the DNA sequence that suppresses promoter activity? Silencer, yes, right. So why do we even have to suppress something? Your aim of that is to enhance, say for in case of enhancer, okay, understandable, it is to enhance the promoter's activity. So why do we have to suppress promoter activity? How does it help in any case? Why do we even need a silencer? Ushma, are you trying to answer something? Okay, regulating overgrowth. Overgrowth in the sense like overgrowth of what? Cells are or production of metabolite or what does it do actually? Cells. So why do you even want to cause regulate the overgrowth? How much ever more is there, that much is good for us, na? Usma, can you mute your mic? So, yes, to suppress the growth to produce secondary metabolites. Okay, that can actually, that is actually good. And there may be other reasons also. Ushma, can you mute your mic? They reduce the transcription, like they don't let your promoter work further, right? So if you don't want something for a longer period of time, just you have to insert a silencer also, so that they will just go and suppress the promoter activity if it is no longer needed. So it is like a switch, a genetic control to produce something up to an extent, and after which you don't need it, kind of that. So whatever Sashank told, it is right, and there are other ways also. In next class, you guys let me know. Find out about what is the role of what are the other roles of silencer in suppressing promoters activity. One answer is whatever like Sashank told, like to suppress the growth to produce secondary metabolites. So one more thing. So do you think just to suppress the growth and to produce secondary metabolites, we are going with silencer? There are many ways. So you guys note down, and next week we will discuss about that. Any of you should answer. Any one of you. So, dash is a small circular double-stranded DNA molecule that can replicate sorry, independently in most bacterial species. Plasmid, yes. Okay, now let's talk about plasmids. Apart from whatever being explained here, what generally plasmids are being used for? RDNA technology, okay, RDNA, which is recombinant DNA technology. Bacterial conjugation, okay. In nature, it is used for same purpose. We have adapted this bacterial conjugation into transformed that into a technology. That is what is RDNA technology. So whatever Shashank told is correct. So they generally are used as vectors, right? So these plasmids are vectors. What are vectors? Sorry. Yes, it is your carrier of your gene of interest. And so, okay. So, what are the qualities or you require in a vector? If you want to select a plasmid as a vector, what are the qualities you will look into? So, you want to, yes, self-replicating. Okay. What else? Self-replication is one. What are the other things you need in a vector? It should have an origin of replication. It should have a selection marker. What else it needs? It should have an origin of replication site. It should have a selection marker. What else? What else you guys need? How many copy numbers you need? You need a higher copy number plasmid or lower copy number plasmid? And how do you even select high copy number? Okay. So you go for a higher copy number plasmid if in case it is used for which purpose? Okay, before all these things, how many vector types are there? One is your expression vector, the other one is what? Sorry. Yes, one is your expression vector, the other one is which vector? Expression vector and cloning vector. So what is the purpose of this expression vector? It will express whatever gene you are putting inside it and give you the product. What does cloning vector do? Whatever gene you are putting inside, it will clone it, make it as multiple copies and give it. That's all. So, so in case, say for example, Shashank told that higher copy number plasmids you would prefer. So higher copy number plasmids will be present in what? Which vector? You want high copy number in cloning vectors. Yes, in expression vector because you need to express that particular gene of interest in more. Like it's yes, you are absolutely right. To get higher yield, you need, if in case your objective is to get expression of that particular transgene, you have to select a vector or a plasmid which has high copy numbers. And if you are going to just clone the cells, you are just going to clone your gene, then you can go with low copy number plasmids. So I'm telling copy number, copy number. So what is copy number? What is copy number? Number of plasmids present in a system. There is a, that is not just about the number of plasmids present in the system. It is a ratio, right? It is a ratio of number of plasmid DNA to the number of what? It is the ratio between your plasmid DNA and chromosomal DNA. With this much amount of chromosomal DNA present, how much is my plasmid DNA? So it is a ratio. It is not a number, it is a ratio. Okay. So that's all. These are the other things. There are no other things which you will look into for selecting a particular vector, right? Do you think size matters? Size of the vector matters? Does the size of the plasmid matters when you are doing a transformation experiment? Yes. So, so do you guys remember about anything about conjugative and non-conjugative plasmids? What are conjugative plasmids and what are non-conjugative vectors? Conjugative will have specific genes which will aid the plasmid's transfer on their own. The non-conjugative won't be able to transfer. Yes, gene and phyli, yes. Phyli with the help of MOB gene. So, smaller plasmids, they will be present in case of non-conjugative. What are they? They are non-self-transmitted. They are very small and they have relaxed DNA cell like control, replication control. But in case if you are going with your large sized plasmid, they will have very stringent DNA replication control. Very tightly regulated gene control will be there. They will always be present at low copy number. Okay, don't forget these things. In case if you guys want to design some vector, keep in mind about the different things which should be present in your vector, which is the origin of replication is important, restriction sites are important, then what your selection marker is important, which will aid your selection of your transformed organism or cells. Selection marker, then copy number is important, size is important. These are some of the things you have to look into before selecting your vector. And also you have to keep in mind on what regard you are going to use this vector. So if it is expression of a transgene, you will go with an expression vector. If it is just cloning your or multiplying your genes, then you can go with cloning vector. Okay, I think we were talking about this for so long, so you can answer this question. Dash are vectors which are used for making millions of copies of DNA segments. Yes, cloning. You are right. Shashank, you are right. So cloning vector. So we were discussing already this in the previous question. So no need for further explanation at this. So the onc region of tDNA is responsible for what? So this one is about your agrobacterium mediated transformation. Yes, the answer is A. So can what is tDNA and is it present in both agrobacterium and both agrobacterium rhizogenes and agrobacterium tumefaciens or only present in tumefaciens? Only tumefacients are. Don't confuse with your TA plasmid and RI plasmid. tDNA is present in both. tDNA is nothing but transfer DNA, right? Am I right or wrong? TA plasmid is different, RI plasmid is different. This is from lysogenes, this is from tumefacients. This will have a plasmid and this is also plasmid. Yes, you were talking about TA plasmid. Yes. So tDNA is there in both. So tDNA is just the transfer DNA. That's why it is. named as tDNA. So what and all this tDNA will comprise of? Okay, before that we will talk about TA plasmid. So TA plasmid is there. What are the regions, major regions you can talk about TA plasmid? You will definitely have a tDNA. What else you will have in your TA plasmid? Apart from whatever we talked about it earlier, there is definitely going to be a replication site, everything, origin of replication, everything. Apart from that, major things. Say for example, tDNA is for transfer. So what else is there? There are four different regions, right? Left boundary and right boundary. Okay, left and right boundary regions will be there of the tDNA. So it'll be somewhat. So what else will be there? This is your transfer region. The other is where your replication happens. Replication part is there. Conjugation part is there. What else is there? Your virulence is there. Whatever which is helping ESPR factors. So leave now the TA plasmid. Let's think about your tDNA. Apart from your boundary sequences, what are will be there? What are the genes? One is onc gene. Somewhere we will keep onc gene is there, which is responsible for synthesizing your phytohormones. What else will be there? Onc gene we know, LB we know, RB we know, opine synthesizing gene. What is that? OS gene. And your virulence factors, vir gene. Okay, so now this tDNA is now getting transferred into your host, which is your plant. So, what are the so in case if I'm going to introduce a new gene and going to into this TA plasmid or TA plasmid. So, which region of the plasmid I have to insert my gene of interest? Where? Which region of the TA plasmid I have to insert my gene of interest so that it will go and bind inside the host organism's genome and start producing my or producing my gene of interest compound. Between your right border and left border. Okay, so Shank is telling that between LB and RB, or you can keep it near your where somewhere between here you can keep. That will be the best option. But you have to. remove certain genes, na? Otherwise, what will happen? If you allow it to transfer as such, what will happen? Your TA plasmid and like your TA especially is bulkier, right? It is very big. Think about it. So between the L and B, like LB, which is your border sequence, we can keep, or we can keep it near where? Any of the borders near it, you can keep. So not always in between your. It's either you can keep it like this, or if your L is it here, you can keep, or here you can keep, or at the middle also. So it should be between the LB and RB, like Shashank said. That is the ultimate aim. So we have further few more questions only. So presence of polyvinyl alcohol enhances the uptake of DNA by cells or protoplasts for genetic transformation. Whether the statement given here is true or false. Presence of polyvinyl alcohol enhances the uptake of DNA by cells or protoplasts for genetic transformation. The answer is true. Any idea why this answer is true? Like how does it? No idea. You guys check the answer for this question. Next week I will ask about this question also. Laser-induced DNA delivery is a method of indirect gene transfer. Whether the statement given here is true or false. Whether it is direct gene transfer or indirect gene transfer. It is direct actually. So the answer for this question is false, right? In laser induced DNA delivery, what it will be done? The laser will directly transfer guided through a guided laser beam. Your vector with the transgene will be directly transferred to the host organism. So the next question is, plant cells are more sensitive to herbicides than to antibiotics due to their specific mode of action towards plant cells. Is this statement true or false? True, yes, they will be more sensitive to herbicides. So, is it advised to generally do a selection with the help of antibiotic or with herbicide? What do you think will help? If you keep a selection marker of antibiotic or herbicide, what do you think that will work fine for plant cells? Okay. So, but plant cells will die if you put herbicides, na? That's why you're keeping a selection marker like that. Like, but there are both ways. People keep antibiotic like hygromycin, ampicillin, etc. also, and herbicides also. Unless it is not causing death of your cells, you can keep any among this. Okay. So the last question: hairy roots can grow in a plant hormone-free medium under in vitro conditions. The answer to the question is true or false. What would be the answer? Hairy roots can grow in plant growth hormone-free medium under in vitro conditions. Is the statement true or false? True. Yes, they have the capacity to grow at a faster rate. So, like compared to a normal cells, they normal roots they grow up at an absolutely faster rate, and they just don't necessarily need any plant hormones because they themselves will be have higher expressing phytohormone genes, so they will not require any phytohormones in the media additional to your other components like sucrose and micro and macronutrients. So they don't need any hormones for their growth. So that's all. We have come to the thing. Yeah, that's all. So we have come to the end of this session. If you have any doubts, you can ask me. Have you guys submitted your last assignment? Week 6 assignment is done or yet to submit? submitted already. Okay. So if you guys have any question, please ask me. Otherwise, we are done for the session. So you guys can leave. So students, we have come to the end of the session. If you have any doubts, please mail in the discussion forum, mentioning the date of the session and mention it as live session. Um, Thanks for joining. We will meet